Thank you very much. Thank you for inviting me. Andy sends his apologies, and he really wants to be here today to share all the work that we have done so far. So what I'm going to talk about today is discuss with you the new drugs that are on the horizon for neuroblastoma, and going to talk why new drugs are needed and why is drug development needed in neuroblastoma. What are the new drugs available on the horizon? Which drugs we are interested and we want to develop for neuroblastoma. And I'm going to talk about a little bit on Beacon neuroblastoma study that probably most of you have heard. And then what is the future envisioned in drug development in neuroblastoma. So why are new drugs needed? Well, the short answer is because we don't manage to cure all the children with neuroblastoma. And then the long answer is, well, we want to introduce new drugs into the frontline therapy that together with radiotherapy that we have heard today and immunotherapy that we are hearing more and more, we can improve the rate of cure for children with high-risk neuroblastoma when they are diagnosed. But equally, we want to improve the treatment for those patients that relapse of neuroblastoma. So, in few words, we want to offer more options to parents and children. This slide shows how the number of new trials have increased over the past few years. If we look at before 2007, there was almost a flat line. But after 2007, when the regulation changed at the European level, that if you are a pharmaceutical company, you want to have an approved drugs for a particular disease in adults, if it's relevant, you need to have also what we call the pediatric investigation plan. So pharmaceutical companies somehow have to commit to do also studies in children. And we are working on that. And as you can see, the total number of studies in red has increased much higher compared to 2007 today. And also we have a higher number of molecularly targeted agents. And I want to draw your attention also on the green part is the first in child. We usually want to be super safe and we want to have enough data on adult study before going to children, but we are more and more seeing that doing studies in children is safe, as safe as adults. So we want to move towards first in child studies and not wait until all the possible studies in adults are done. So after a drought of new drugs have been available, now we are really having a monsoon of new drugs. And we need to be aware of this because pediatric cancers remain a rare disease, and we do need to join forces in order to have, yes, drugs, but also to do clever study altogether. So what is the process of drug development? Well, it's a very, very long process. We start with preclinical data. We look under the microscope. We see the molecular level. We try to understand how the tumors work at the very, very specific level. And then we suddenly have a drug to target that particular characteristic of the tumor. And then we need to do a phase 1 study. Now, phase 1 study is merely, unfortunately, only to look at toxicity, to be sure that we are doing the right things and we are not harming our children. At the same time, looking at what is the dose that we will have to use later on. And then we'll move into a phase 2 study where we enroll more patients. We allow more patients with a particular disease or a particular subtype of disease, and then we treat with that drug and see if it works. And then if it works, then we will move in a phase 3, which in pediatrics is frontline treatment. Now, the problem is there is a very high attrition rate of drugs. So 95% of all the work that is done preclinical then eventually doesn't reach the phase 1. And 95% of the drugs that reach the phase 1 don't reach the phase 3 for several reasons. It might be toxicity, it might be lack of efficacy, it might be because the drug is no longer available. So how can we improve this? Well, if you look at the activity response, the probability of a patient enrolled in a phase 1 study to benefit from that study, from that drug, is 10%. If we look at the new drugs, what we call the clever drugs, the clever bullets, that target specific characteristic of a particular tumor, then the likelihood of response is 4%. And this is because the tumors is very heterogeneous and there are several characteristics that matters in a particular tumor. But if we combine a molecular targeted agent with the right patient, so if we know about the characteristic of a particular patient, a particular tumor, then the likelihood of response is 50%. So why has been the introduction of new drugs into clinical been slow? Well, there was a low number of drugs available for investigation in children. Often the clinical trials were not optimally designed. We don't always look at the best drug or ask a clear question, or we don't select the right patients for the trial. And we often don't understand what's happening in the tumor when we put in such drugs. And we often don't compare with the standard treatment. So understanding what is the real benefit of an increased survival of 10% or 20% if we don't compare to the standard treatment is very difficult. And as we have seen today, the clinical trials take a very, very long time to develop. And this is because of the bureaucracy, of the time, of the safety issues, and all that we have discussed today. But also I want to remind you that pediatric oncology is a small ward. We don't have a very large number of patients such in adult cancer. So we need to probably join forces together. So the importance of doing the right trial with the right drugs and the right biomarker to select patients comes from the adult experience. For the liver in chronic myeloid leukemia, well, the Time published that, well, apparently we are facing a new era where we can actually beat cancer. And this is because if we have a specific target and we combine it with the specific drugs, then the likelihood of response is much higher, and ideally this would reduce also toxicity because the normal cells wouldn't have that target. And there are a number of examples in the adults world. So molecular targeted agents used in conjunction with predictive biomarkers might be the way forward. So we are shifting the paradigm. Before we have the drug that we put into several patients with several cancers, and then we would see which one would respond. And obviously in this case, the rate of response is very low. We want to shift this paradigm. We want to be able to select the right patients for the right drug, and then give the drug that particular patient might benefit. And we want to do this also obviously in pediatrics and in neuroblastoma, to develop better drugs, to develop more effective drugs, and to develop them faster. So we want to follow the success that was in adults and in particular in Gleevec. So a paradigm shift towards more biological, more hypothesis-driven clinical trials with a better selection of agents. Now we have many drugs for the same target, and we need to select which one is the best. We cannot afford to do a study on every single new drugs that target the same target. We need to be able to select and to see which one is the best. We need to have a good and trustful preclinical model, because as we have seen, all the phase 1 comes from the thoughts that are before the clinical part, and this is the preclinical model, the in vitro and in vivo model. And we think that one of the best models would be the genetically engineered murine models, that, for example, in neuroblastoma, there is a spontaneous onset of neuroblastoma that might recapitulate better what is happening also in human. We need to incorporate the biomarkers. So biomarkers are these small characteristics of the tumor that would help us to select patients and see which one would respond better. And we need new trial design. We have seen how long it takes to build on a trial. So if we can do this effort once and be able to test more drugs, it would speed up the process for going from a phase 1 to a front line. And then, as I said, we need a pediatric drug development network in Europe and also across the Atlantic. So the interaction between all the stakeholders is very, very important. So academia, such as clinicians, pharmaceutical company, regulators, and obviously you. So what are the new drugs on the horizon for neuroblastoma? Last year, many of the neuroblastomatologists, both biology and clinicians, met to try to have a prioritization list of the new drugs based on what is the biologically rationale to target this particular target in neuroblastoma. But equally, what is the best drug? If we got a drug that could effectively target this target. And we came up with a list of new potential targets. The first on the top was ALK, but also Aurora kinase, the survivin protein BIC5, CHEK1, MDM2, and many others, based on the preclinical evidence, but at the same time, the availability of the drug. And this really much changes according to what's happening. So if today we have a better drug for another target, then that one will get the priority. We need to be able to design short, rapid dose escalation trials to find out very fast which is the dose that we have to use in pediatrics, and then move into expansion part of the study where we enroll patients with the same disease, with the same characteristic, to see whether the drug is effective or not. And one of the methods would be a multi-arm, multi-stage study such as Beacon, which I'm going to discuss later on. And again, it's very important to understand what's happening in the tumor, but we cannot do it without a tumor sample. We need to understand what are the molecular characteristics of neuroblastoma tumors in order to design personalized, what we call precision medicine. So we have different phase 1 study potentially already open and that will be opening soon, as we see here with all these very strange names. But all of this effort is to then move towards testing this drug in a relapse setting in a Beacon study, or better, what is happening after Beacon. Beacon study is a study for relapsed neuroblastoma patients, and I'll talk about it. But it's important, as I said again, the molecular characterization, and this is why ALK has been a very successful story. We have ALK is the first molecular predictive biomarker that has been used in neuroblastoma, and from crizotinib study, then now we have other different agents such as LDK378, novel agents, but also we are looking at combinations, because we think that the way forward is not just one drug will cure definitely one disease. We need to target the disease from different angles. And this is a list of trials that are open or opening in the UK. So as you can see, there are many phase 1 and phase 2 studies for children with relapsed neuroblastoma, from the immunotherapy to radioimmunotherapy and molecularly targeted agent. And these have opened or will open between 2013 and 2014. But also we have a number of other targets and drugs that we are now working on, and they will hopefully open in the next few years. So we're now over 20 new drug trials in development, and you can read the list. I don't know whether this will tell you anything, but it's basically a way of targeting the different characteristics of a same tumor that we call neuroblastoma. And combination study, we think, is the future for molecular targeted agent. So which drugs we want to develop? Well, an example, we focus on identified molecular abnormalities that initiate and maintain the tumor. And we know most of them, MECAN, ALK, but also we target p53, which is another important protein or different pathway or other molecules. And then we approach this through different strategies. For example, targeting the MECAN, which is the most known target, but also targeting ALK or the angiogenesis of the tumor, for example. And this is an example of how target MEC. We saw a similar picture before, so this is the cell membrane. And this is what's happening inside of the membrane when a growth factor attaches to one of the receptors. There are many pathways, and there are much more than these just two. And this will induce proliferation of the tumor. Now, if we manage to stop one of these or more of these, then we will hopefully have a response. And ALK is a little bit of the paradigm of what's happening in children, what we want to happen in children. We identify ALK as a major familial neuroblastoma predisposition gene, but we saw that ALK is also expressed in sporadic tumors. In 10% of those have mutations and 4% have amplification. So we developed strategies to target ALK in a specific way. But as I said before, we think that combinatorial approach is the future. And this comes from a preclinical work that has done in Louis Chesler lab, where in using a murine system, here you see the volume change from day zero, the volume of the tumor upon treatment. So if we don't use anything, then the tumor goes up. If we use the ALK inhibitor, degree 1, we have a delay in tumor growth, but the tumor remains basically the same, as well as if we use another pathway target, that is the mTOR inhibitor, taurin 2. But if we combine them together, then we will have a much better response. And this is a survival curve, and as we can see, the red one, which is the combination, is much better than the other. And we can do MRI scan on these mice, and we can follow the tumor response. And as we can see, the combination of the two molecular targeted agents has a better outcome. So what is a biomarker? As I said, it's something that will tell us, A, whether the tumor will respond. So what's the likelihood to respond to a particular treatment? But at the same time, it's important to see if we use a targeted agent, whether the target has actually been hit. And that's how we can actually see whether if there is a failure in response, whether it's because the drug is not good enough, and we need a better drug, or whether we need a better target. So go back to the bench and have another hypothesis. But obviously it's very difficult for children to have many biopsies and see what's happening in the tumor. And that's why we are developing new ways of looking inside the cells without actually putting patients under many biopsy. And we look at the bone marrow or circulating tumor cells. So we try to isolate the neuroblastoma cells from the bone marrow or from just the tumor, from the blood, and see what pathways are inhibited when we use a targeted agent. Or even moving towards less invasive procedures such as an MRI, looking different techniques to look at different aspects of the tumor and how these aspects changes upon treatment. So Beacon neuroblastoma study is a very innovative study because it's a phase 2 randomized study that looks at 4 different treatments within the same study and look at 2 different hypotheses. So we want to answer 2 questions from this big study, and these: which one is the standard treatment for relapsed neuroblastoma? Is it temozolomide alone, or is temozolomide plus irinotecan? We have different results from the phase 2 study that have been published, but we don't know from randomization which one is actually the best or which one is the less toxic. And at the same time, we had the possibility to evaluate a new targeted agent, bevacizumab, that might have a role in neuroblastoma. So from this study, using a low number of patients, we would have to answer, and this is a way forward to speed up drug development in children. So what is the impact longer term of Beacon? Well, it's very important because, as I'll show you, Beacon has created an international trial network infrastructure that will allow to go on and do what we call the Beacon 2, see what's happened after Beacon. And at the same time, somebody has defined Beacon as a living trial, because as soon as we have maybe another possibility, another potential standard treatment, we could incorporate that in Beacon and test it straight away without waiting other 3-4 years to set up a study. And at the same time, in Beacon, we will do what we call the test predictive biomarkers to see if a patient responds, can we predict that response, can we follow up that response better. And this is the network that we have established to do Beacon. We started from the UK and the very help of University of Birmingham, with the chief investigator being Andy Pearson. But we have obviously recruited 8 other European countries with 32 sites for all the children in Europe, most of the children in Europe with relapsed neuroblastoma, to have the possibility to be enrolled in the trial, for them to benefit of new drugs, and for us to understand what is the best treatment for these children. So what is the future? Well, we think obviously that working together is better. And we have strong links with CEPAN, which is the European network for neuroblastoma, new link with German group, but also new links with Nantes and with the Children Oncology group. And we are very much embedded in the ITCC and the ncca group, which are at the European level, the groups that look at the new drugs and facilitate the relationship with the pharmaceutical company, but also with the regulators. We want new trial design, what we call the octopus or drop the loser. Somebody argued that it's better to call pick the winner. So we have different drugs, new drugs that are tested against what is the standard of care. And we don't want to pick the winner. We don't want to pick the one that actually gives us a response rate of 5% more. We want to drop that drug because we don't think that that's one is interesting. We want to have a very big improvement, like 20%. And that's why we want to drop the loser, not pick up the winner. And this will facilitate a rapid progress from the phase 1 study to a frontline therapy using stratification of patients by the ones that I described before, the predictive biomarkers. See which patients might benefit from a particular drug and use that frontline to improve the survival. So what are the answers to the point that I did before? Well, low number of drugs, but we have now improved our links with pharma and regulators, and we have regular meetings with them, so to understand what are the needs of the academia, what are the needs of the pharmaceutical companies, and the needs for the regulators, and do this together to speed up the process. Do better drugs that are more biologically driven, more clever studies that would look at predictive biomarkers and pharmacodynamic biomarkers to see what is the response that we get into tumor when we give what we call the clever drugs. And at the same time, it's very important to do a randomized trials, because otherwise we will bias. We might select the patients that are better for a less toxic drugs or a more toxic drugs according to what we think the patient might tolerate, but that might bias the final result. So it's very important to randomly select the patient for the different arms. And yes, clinical trials take a long time to develop, and that's why we want novel and efficient trial design, so that we don't have to wait years to have one trial, but we can start enrolling new parts of the trials in the same setting. So in conclusion, a large number of drugs are becoming available for neuroblastoma, but we need to be clever, and we must rationally select the best ones for children and for children with neuroblastoma. We think that enrolling patients in clinical trials is the way forward, and there is much progress made. And Beacon neuroblastoma indeed is a very much major step forward, especially because it has built up on an international network. And obviously the ultimate goal is to have better drugs to cure children with neuroblastoma. And with this, I want to thank all the Royal Marsden group that has done and is doing drug development. The list when I started was very much up to here, and now we've got many other names that have joined. And Louis Chesler Lab that is dedicated all to neuroblastoma. Michelle Garrett that mainly leads on the pharmacodynamic biomarker, and Simon Robinson that leads on the preclinical imaging. And obviously the links with University of Birmingham and Cancer Research UK, and but also all the links with SciUPenn and the ITCC, and you for your attention.